Now I request Professor, Associate Professor is still a Professor, sir. Professor Avatharam Ganivada from University of Hyderabad. Please be seated, sir. So,
respected HOD Madam and Principal BOS of the Chemistry, and Kamal Sir, Lalita Madam, Brahmanandam Sir, and senior faculty members and dear colleagues and my dear participant. On behalf of the organizing committee, I am extending a warm welcome to all participants to this two-day national workshop on AIML driving drug discovery. It is my privilege to introduce our distinguished speaker, Dr. Avatharam Ganivada. Dr. Avatharam Ganivada is an associate professor in the School of Computer and Information Science at the University of Hyderabad. He obtained his PhD in Computer Science and Engineering from the Indian Statistical Institute, Calcutta, under the guidance of Professor Shankar K. Pal, and holds an MTech in Computer Science and Technology from the University of Mysore. Since joining the University of Hyderabad in 2017, he has been actively involved in teaching and research in areas such as deep learning, feature of systems, neural networks, feature selection, and medicinal image analysis. The publications in biometrics, ultrasound imaging, gene expression analysis, and AI-enabled drug discovery research. Dr. Ganivada is currently associated with national research projects funded by ANRF and other agencies, including AI-ML-based approaches to disease predicting and drug discovery. He has authorized a Springer monograph on and he published several research publications in reputed international journals. Under his guidance, 3 PhD degrees are awarded and a few students are pursuing for their research work. Today, Dr. Avataram will be delivering in a lecture on physics of loss based deep learning methods, medicinal images, segmentation on Indenopyrazole. We are truly honored, sir, to such a
This is not working, no? So thank you, sir, for introducing me. So presentation is fuzzy of loss based deep learning methods and applications in medical image segmentation and classification of protein kinase inhibitors. So in this talk, mainly I will discuss some sort of CNN architecture. So it's a deep learning model, basic model of deep learning, and some applications, not some, one application and some results. And now recently we have published with my PhD student. So that work was published in computerized medical image and graphics. So very recently, last month, it was available online. And that title of the work is Fuzzy of Loss Function based deep learning for medical image segmentation. So that I will explain a little bit. I will explain. So the main idea and how we design that model, and I will explain, and I will show you some applications in medical image analysis. So then I will show you some medical image results, segmentation results, so which are the part of the same work. And finally, what I will show you that different AI models, how we apply to that protein kinase inhibitors classification, right? So we all know that biochemistry part and molecular active molecule and inactive molecules and how do we apply that models, JA models like classification models, and how do we classify those active, inactive molecules. And before that, how do we derive those molecules, that I will show you a little bit. So these are the outlines of my presentation. So let us start this on architecture. I want to show you this CNN architecture, and this is the basic model in deep learning. Many people, you all talk about deep learning, right? So this is the basic architecture. You can see that this is input, and at the end is final output. So intermediate, there are many layers. So many layers. So look at here, convolution layer, pooling layer, convolution layer, pooling layer. Just I have shown only 2. So like this, there are many. So depending on the problem, we increase all the layers. So one by one. So finally, fully connected layer. So whatever the features we extracted from this convolution pooling, so we put it in one layer, that is fully connected layer, and initialize some weights and classify it. So that is mainly CNN architecture. Now if you look at independent parts, like each and every part of this model, so what is convolution, what is pooling, what is fully connected, and what are the mathematics we apply in this model. So those things we will see. So convolution is nothing but, for example, you have an image, so you take a simple kernel like whatever kernel, there are many kernels. So we select one kernel, that is 2 by 2 kernel, and that is the image. So 1, 2, 3, 4, 5, 6. So now what we do, just do convolution. Now you select 2 by 2 matrix and kernel matrix, and from that image you select only 2 by 2 matrix. Now do it convolution, like simply product of each individual pixel with each individual value in the kernel. So finally you will get, so since it is a 3 by 3 matrix, 3 by 3 image, and you have a 2 by 2 kernel, finally you will get 2 by 3 output. So this way we extract features from the any given input image. So this is a simple, so just to understand the concept of convolution, I have shown this slide. So this is the convolution between given input image. So the image could be anything, even if you can take any image from your biochemistry also, or any image, medical image analysis. So I will show you some applications so that you can understand. So this is the convolution operation. So mathematically, how do we do that convolution between given input image and kernel function? So the way I explained in the previous slide with some example. So this is the mathematical notation. This is the height and width dimension, and that is the kernel function. That is also height and width dimension. So in the previous slide, what I explained, just I have taken 3 by 3 image. So kernel is 2 by 2 matrix. So now we do convolution. Finally, we will get that output convoluted image. So why do we do convolution? So given that image and we apply convolution operator, why do we do that? So mainly these are the important points I mentioned here. One is parameter sharing, and the second one, so when you apply kernel matrix through whole image to extract some good features, salient features from that particular image, that features are useful for classification. So that is the important thing. And another one is sparsity of connections. So we just do some sparsity of connection between input and that convolution kernel function. And so mainly less number of connections are required, as output value depends only on a small number of inputs. So these are the basic things which are required for convolution. So what is activation layer? So we have done convolution. Now what do we do? For every convolution, we have to apply activation function. So in mathematics, there are some mathematics equations like sigmoid function, ReLU activation function, and tanh function. These are all simple mathematics functions. Whatever that output image which is obtained by convolution, so we transform it in a different domain, that is called activation, by applying activation function. So for example, you get the convolution in one particular domain, like a linear domain. Now we transform it to non-linear domain. How do we transform? Just by applying sigmoid function and ReLU function, tanh function, any one of the functions we have to select it depending on the problem. So this is the activation layer which is there in CNN architecture. So if you look at that diagrams or graphs, so if you take that one sigmoid function, that is the equation, so 1 by 1 plus e to the power of minus x, you look at the curve, it lies between 0 to 1. Now you take the another tangent function, that is hyperbolic tangent function, so look at the range, minus 1 to 1. So similarly ReLU function, so it is 0 to 1, only positive values, we are not going for negative values, so like sigmoid. So like this there are different functions, exponential ReLU function and leaky ReLU function. So generally we use ReLU function or sigmoid function in most of the cases. So for any particular problem we always use ReLU function. So deep learning means you always use ReLU function or sigmoid function. So every convolution layer or every activation layer we just add this function to transform the output of activation convolution to nonlinear domain. That is when you apply this activation function. Once you do that, what we do? So we have that nonlinear domain, that nonlinear output. So we have to reduce something from the, after doing convolution, so we have to reduce something from that image, output image. So then we apply some different operators like max pooling operator, minimum pooling operator, average pooling operator. There are different functions. So you select any one of these functions, any one of them, not all. So you select any one of these functions and do pooling. Pooling means our objective of pooling is to minimize the actual convoluted image. Size of the image we have to reduce it. So then we apply this pooling. Just I will show you one example. How do we do that? For example, this is your convoluted image. After doing convolution, after doing activation function, after applying activation function, this is your final output. After those two operations performed on a particular given input image. So this is your image. Now what we do? We have to reduce unnecessary things. We have to remove from that output image. Now you select it, the 2 by 2 image. So you take max pooling. The 20 is the maximum. Again you select 2 by 2. So 30 is the maximum. So again 34 out of this 2 by 2 matrix. So what is the maximum? 112. So like this we have to select only that important values by doing max pooling. So if you want to apply average pooling, so what we do, just take the average of the first 2 by 2 matrix, second 2 by 2 matrix, third and fourth. So then average pooling. If you want to do minimum pooling, so you select only the minimum value of in the particular image, like 2 by 2 image. So here minimum is 8 and the second matrix minimum is 0 and third matrix 34 and the last one is 4. So any one of the max pooling operators we have to apply on that particular image. It's all depending on the problem. So what we do now, fully connected layers. Once we perform convolution and activation, applying activation function, after doing fully connected layer, pooling operation, after performing pooling operation, finally you will get one output. This is for one convolution. So this kind of convolutions we will do many things. Many convolution operators we will apply on a particular image. So our objective is to select different salient features. So those features finally we will make it into we will feed it into the fully input of the fully connected layer. That is the fully connected layer. In the diagram I have shown that fully connected layer. So this fully connected layer, like any neural network model, so just to do for classification. So this is the fully connected layer and essential component of convolution neural networks. So finally what we do, with the help of fully connected layer, just we do classification. You take this image, simple, I have mentioned one tiger. So what we do, when we apply that all the convolution and our activation functions, and so you will get feature maps like this. So first one is L1, one activation function, second one another activation, third one. So different activation functions when you apply, you will get a features. So this, given this a particular image with a particular animal, so we extract features. So when you apply different activation function, you will get different feature. So that's the image I have shown there. So overall example, if you take this simple image, so that is a 2 by 2 matrix or single 1 by 2 single one dimensional array. So 1, 2, minus 3, 4, 5. So your kernel is also a single one dimensional array, not 2 by 2. We are not taking 2 by 2 matrix or 2 by 2 array. We are taking only one dimensional array. Given input as a one dimensional array and your kernel function is one dimensional array, do convolution the way I explained. So we do convolution, you select 2 points from this input and 2 values 2, 1 and you will get 4, 1, minus 2, 13 like this. A simple convolution, I have explained what is convolution result. So then we apply max function, that is activation function. Once we do convolution, we apply max function. We select out of these what is the maximum value, 4, 1, 0, 13. So that is the output of this activation function. That output again we will send it to this one pre-processing, right? So again we do convolution layer, pooling layer and applying, finally you will get fully connected, that is the last one, fully connected layer. So this way we have to repeat it several times, convolution, max pooling, activation, max pooling. So how many convolutions are required, that we have to select initially in any convolution neural network and do the same process several times and finally we extract the features from that given input image. So mostly in deep learning we will work on only images, mostly. So we can also apply some numeric data. So then we have to change the architecture like this, single dimension. So if you look at some applications, image classification, one application. So just now I have shown some image, right? So tiger, that is an animal. So we can classify that one. So we can also apply video analysis. So we can also image recognition. So NLP, natural language processing, also we can apply this. CNN architecture we cannot apply. There are some other models like recurrent neural networks and all the things. Those sequential data, because natural language processing is always works with sequential data. So there we will apply NLP, this recurrent neural networks. So some time series analysis also, recommendation system. In bioinformatics there are many applications, and even that biochemistry there are many applications. So drug discovery and also that initially I have shown, right, Kinsey inhibitory classification. There are many applications. 1 or 2 applications we will discuss. So we run that simple CNN model on this particular image data, that is MNIST. So here there are some images, 20 by 28 grayscale images, and we select 60,000 samples, like images 60,000, training data and test data 10,000. Now we classify based on CNN model. So how many are classes here? There are 10 classes. So that means we classify that image MNIST data set. We have a 10 classes, and each class, what is that, we have to classify using CNN. So when you apply that, I have mentioned only 3 different models. One is CNN with 3 convolution, 3 pooling layers. Similarly, VGG system, this is also a deep learning model, right? So ResNet, this is also a deep learning model. Only the difference is we increase the different number of convolution layers and activation layers, right? So his VGG system means there we perform 16 convolutions and 16 max pooling and different other operations we perform. And ResNet, there are 50 convolution layers. So like that we increase. So finally what here, our CNN model with 3 convolution, 3 pooling layers gives you better results than the other 2 models. Even if you increase number of convolution layers and number of pooling layers, so you may not get the actual results or good result. Even less number of convolution layers, less number of pooling layers, you will get a better results than the other models. So that's why I have shown some results here. And this is the basic architecture for any deep learning model. So with this model, we extend our work into different models like segmentation model. So many models. In this work, we have used almost 8 models like UNet architecture, UNet++, MobileNet, ResNet. There are many different models are there. Mostly we apply for medical image segmentation, right? So in CNN model, what we have discussed, we just architecture I have discussed. But when you talk about training any particular in neural network model, it requires a loss function. Even CNN requires a loss function, or any deep neural network requires a loss function. So in our work, what we have designed, we designed a new loss function that is called fuzzy rough loss function. Right? We use fuzzy concept, we use rough set concept. Right? So instead of a general loss function or any conventional loss function, so there are some optimizers like Adam optimizer, different convex, concave loss functions. So instead of that, what we have designed a fuzzy rough loss function, because so you look at that image, input image, you see that mask tumor part, we can't see that tumor part properly. There are a lot of noise in this image. So background part and that particular tumor part we cannot identify properly. So in that case, what we do when we apply fuzzy, so we can change that background, we can identify that particular tumor part clearly. So look at that predicted mask and ground truth, that is the actual ground truth particular image. It is available online. So the data sets are also available online. So we select the data set and we select the same ground truth for that particular image. So what we do, we design a new model according to that particular data. So that is what we have done. So we designed a model. So what we have done here, you look at here different fuzzy set components. So in this fuzzy set component, we designed a new similarity measure. So the steps to design that new similarity measure, like so statistics we have applied like mean and variance. So some distance calculation, membership value, so influence value, fuzzy similarity. This is our own model. So our own fuzzy similarity equation we have defined. So this initially we have published in 2019. One Machine Intelligence Conference was organized here in Hyderabad. So there we published that similarity relation, and the same concept we have used in this medical image analysis to define a fuzzy rough loss function. So that loss function is mainly based on different fuzzy rough concepts. So one is lower approximation. Mostly you all don't know how to define lower approximation and how to define upper approximation. A lot of mathematics are required to define those functions. And so finally you will get one lower loss and upper loss and this fuzzy loss. So from this fuzzy similarity, we just you combine or integrate all the things based on one concave function, convex function, and finally we have defined FRS loss. So this FRS loss function, just what I am saying here, we just in what way we can define a new thing, just I am showing here. In this model we have defined a loss function, and you select any model, you can define your own way. So that should be should be suitable for your work, or should be suitable for your data. So you can change the model architecture. So the way I have shown like 3 different models like CNN, ResNet and VGG. So what they did, they changed the architecture. They changed something new. They added something novelty in that architecture, and they have shown some results. The way we did, the way they have done in different way, we brought some fuzzy concept in that model, and we designed a new model like this. So the basic mathematics which are there, what we have used, so I am showing in this slide, like this is the lower approximation equation and upper approximation equation, and the last one is fuzzy similarity equation that we have defined. So different equations, and the loss function, finally we use a convex function, and we have defined the loss function. All the mathematics we have derived all the things here, and this loss function, what we did, we apply some different deep neural networks, not a single neural network. So I have explained, right, like UNet architecture, Signet architecture, there are many models. So we selected 8 models out of that, and for all the models we use this loss function for all the models. And how that model works, just based on our loss function we have checked, and we have selected some different ultrasound images, data sets freely available on Google. So we have selected, and what we did, so when we apply this, the proposed loss function, the last one, So it gives you a better results in terms of two different indices. That is called, one is dice loss and another one IOU, union intersection over union. So these are the two indices which generally we use in medical image analysis. So we provide results, or we evaluate our model's performance using these two different indices like dice and IOU. So you look at these diagrams, so everywhere our model performs better than the other models, like other loss functions. Like, so if you look at the first row, we remove that fuzzy component, so in that loss function, so the results are not so good. And if you look at the second row, second column, so we remove that membership function. So initially we remove that similarity, and the second row is we remove membership, and the third one is we remove rough set. So if you remove one component from that loss function, how that results are, just I have shown here. So and finally, if we add all the combinedly or integrated all the things in one particular loss function, that is the proposed one, and gives you, and you look at that last column, so accurately define, accurately detect the region in that particular image. So I have shown that input image, right? So initially that ultrasound image. So what is our objective here? To detect that region, that tumor part. So we have used different chest CT, that is one data set, and Kevisar sigmoid, and BUSI, that is breast ultrasound image data, and skin lesions, brain MRI. So what we did here, just we have seen that batch-wise training, and we have seen the training time and all the things. So this is the graphs we have shown. So even if you take that dice and IOE values for different loss functions and different images, this is the original image, and this is one loss, BC and dice loss, different Strauss loss. There are different loss functions. We have compared with all the loss function, and the last one is proposed one. So that is FRS loss. So when you compare all, like for different images, not one single image. The first one is original image, one CT image. Second one is, the third one is brain MRI scan. The last one is lung scan. So different images, and when you take the outputs and when you apply the models with different loss functions, and we see the outputs. So finally our FRS loss gives you the better results than the other models that we have seen here. So this is also still we are comparing with different models like focal loss, Huber loss. So more than I think more than 10 loss functions we have compared with one single model. This is another model, same thing, different images. So we are just simply comparing with all other loss functions. So this is the what is the finally error values when you compare with that loss, means what is the error for different models. You look at that FRS loss, that colors, different colors, that is the lower one, the last one is the lower. So where the other loss function gives you the higher error, so with one particular model. So this is the simple error curves we have plotted in XY plane. So this is the important thing. So how do we decide that which loss is the better thing? By seeing this diagram. So you look at the last one, that is using FRS loss, the error value is very smaller than the other values for different. So this is an application of medical image segmentation. Now our biochemistry application, my student Amit, he is a project student. He started working on this one particular paper, and he has done some work. Just I will show you that work. So mostly here he attended that workshop also. So what he has done, that he has drawn that structure of Indenopyrazole derivatives. That term I can't speak properly. So then what we did, that all the structures we convert into use that SMILE format, convert into different molecules. So from that you extract molecular descriptors using SMILE format. So finally what we do, so use IC50 value to decide which one is active molecule and which one is inactive molecule. So in this way, I mean, he has generated more than 90 molecular descriptors with one single chemical structure. So what we did now, we apply some machine learning models. So out of 90 molecules, we don't require all the 90 molecules to classify, to identify which is active one, which is inactive one. So we remove some unnecessary things. How do we do that? We manually cannot do. So we apply some sort of feature selection or feature redundancy methods. There are some methods. So that I will tell you. So this is the simple structure, drawing structure of that derivatives. So he used different chemical structure with different softwares like chemistry draw, so Marvin. So different softwares we used, and from this, so he has defined that smile format. So using this softwares. So use one particular software Marvin and derive each of the 90 molecules for this structure, right? So that is a smile form. From that he generated different molecular descriptors. That's what he has done. So what is these molecular descriptors? There are some sort of tools again. So he has used those tools from this smile format. So then he finally gets some molecular descriptors, right? So like this he has derived some sort of 90 molecular descriptors, right? But when you do classification or something, when you do classification of that molecular descriptors, so those are all not required. So he removed something. He removed some molecules. How did he remove? So this is that IC50 value he has used, and to decide that which one is the active and which one is the not active, and something is missing, and he has used some missing value. He assigned some missing values in that particular data set, right? So in these moleculars, what we did, he has derived 90 moleculars, right? From that, he removed some sort of unnecessary things using principal component analysis. This is one algorithm which can be used to reduce the dimensionality of the particular data set, right? So, and also he has used some genetic algorithm concept. So actually this is existing work. What we did, we just reproduced the results as it is. We have not changed anything, and we are thinking of designing a new model which can give you better features in this molecular descriptor data set, right? So either we instead of using PCA, instead of using GA, we can use some other model or some other feature selection model, right? And which gives you the salient features compared to PCA and GA. And finally, what is our objective? Improve the accuracy of classification accuracy of these active and inactive molecules. So he has applied PCA and he has applied GA. He has compared these two. So which one is the better? That is the work he has done so far. So, and what are the classification models? These are the existing classification models. You can also apply in your work. One is SVM. So we have not designed any model here. We have not derived any new model or new network. So existing model SVM we have used, and CNN we can take one-dimensional array format and we can apply CNN model. We can also MLP. It is a very popular model, multi-layer perceptron. So these are AI tools. So your data now, once the data is ready, what we do? Just classify the data using these models, right? So for this, to train the models, so the entire data set is not required. We no need to and we no need to train the model with all the samples or all the features. Samples with the features. Features means that molecular descriptors. So for that we use PCA and GA. GA means genetic algorithm concept. So it is a popular concept. Anyone can use that concept, and you can see that Google, there are many Python codes are available. So you can directly apply your data set and reduce or remove unnecessary features from the data. And we can also apply these classification models and see the results. So with PCA, what is that accuracy? Look at SVM and CNN, MLP. When you compare these three different models, so mostly MLP gives you better results, but all are less than 80. So accuracy of means that classification accuracy of MLP nearly 80%, whereas the other two models CNN and SVM it below than which are very below than that MLP. So when you apply GA, so in case of GA, so that MLP classification accuracy is more than 84. And the other thing that when you look at this accuracy, precision and F1, so this is for SVM. What is that? First one is SVM, second one CNN, the third one MLP. So almost accuracy wise, SVM and CNN, MLP almost similar. There is a small difference in point, decimal point. Whereas MLP, so F1 accuracy, that is one measure which is used to evaluate the accuracy of any particular model. So that F1 accuracy is much higher than the MLP and CNN. SVM is better than the other two when you apply GA. So what this GA, the concept of GA used to reduce the dimensionality of the given data set. So we have derived 90 molecular descriptors. We don't require all the molecular descriptors. We remove something. So we apply GA. So GA concept is ready and freely available. So just we have taken some sort of existing concepts and we tried with doing something, we tried to produce some results, right, with existing models and existing feature selection methods. And let us see the results, how the results are, like which model gives you better than the other models. So that model means results in the sense our SVM, it is 86% of classification accuracies, right? So that means active and inactive molecules, classification of these two different molecules, right? So our model is giving 86% correctly. So if you look at that, these are all simply confusion matrices, right, for different models. So the diagonal part correctly identified molecules, either it is inactive or active. So non-diagonals are wrongly identified molecules. So these are some simple concepts in machine learning we apply, right? So thank you. So these are the simple applications of biochemistry, and previous one is medical image segmentation application I have shown, right? And initially I explained a basic architecture of CNN model, right? So these are the basic things. We have not started actual work, so slowly we will work on the actual things. Actual means taking more data sets, right? That we will discuss later. So any doubts? That's all. Those things are 2D images, madam. Yes, 2D images. Yeah, actually here we have taken only 2D images. So yes, whatever the image is there, 2D, no color. Yes, ultrasound image, CT scan, different images. All are in 2D format. Now we didn't take any 3D image here.
Molecule what you show.
Yeah.
What exactly you are trying to do in that? I couldn't get it.
What exactly we are trying to do here? Actually, this we have taken a paper, one research paper.
It's a known molecule.
Yes, known molecule.
Different substitutions or something.
Right. This is the structure. Yes.
Okay. So, what you were trying to find out that which can be better than this something or?
Yeah, actually we are classifying, right? Yeah. We are protein kinase inhibitory classification. So title is protein kinase inhibitors classification. Right? Whatever the molecules which we have derived, so related to protein kinase. So the structure is this one.
Yeah. So which could be much better.
Yes. No. So why once we get that complete data, right? So that's what this is the format, initial structure.
Yeah, but these revision activities are already reported.
Reported.
So this is only an exercise to show whether your model is working or not, or you can predict something which can come out better.
Right. Actually, we trained with known molecules. We split the entire data into training set and test set.
Okay.
So all are known molecules. Right. Now we take the model and train the model, and with test set we classify it, how the model works. We are checking that one. So once you train the model and keep it in somewhere, and you take that unknown samples, unknown molecules, and you can classify it. So for that we are designing a model. Yeah, you can apply. So actually there is no particular size or something. So generally we apply K-fold cross-validation, and there is a technique called K-fold cross-validation. For example, your data have 100 samples with some molecule descriptors. So what we do, K-fold means K equals to 5. You split the entire data into 5 folds. So you select first 4 folds as the training and 1 fold is testing. So repeat the same process until classifying all the right. Yes, randomly we have to select out of 100 samples for split the data. Yeah, randomly split the data and there is no overlapping point in the folds.
It means the scaffold what you are taking should be already validated.
Yes.
Cannot be totally new.
Yeah, at present we have taken known things.
Okay.
Right. So we build a model. For a given data set, what we are doing here is just build a model. So that model is also there, but let us see that model, how that model works for a unknown sample. Right.
Some exercises.
We are just doing some exercise.
So while getting the pooling layer in the CNN, so you have mentioned that there are 3 types: max pooling, minimum pooling, and average pooling. So on what basis do we have to choose that when to use which?
It's all depending on the problem. Mostly we go for max pooling in image data. In image data, we mostly choose max pooling.
What are the use of other pooling methods?
So see, max pooling or minimum pooling or average pooling, what we are doing, we are reducing the size of the convoluted image. Right? That I have shown an example, right? Given that convoluted 4x4 image, finally we get some 3x3 or 2x2. So we unnecessary details we are just ignoring by doing max pooling or minimum pooling or average pooling, any one of those. So depending on the problem, we have to select it. So mostly in image processing, we go for max pooling. So why we go for max pooling? The intensity, you take that higher intensity value, right? So that is the better thing, better one to select. Database, that is the Kinsey inhibitory data. So we have just implemented a paper, only one particular paper, what the data they have used and what the formats and what the softwares they have used. We reproduce the results, nothing else. We have not done any new thing, right? So that paper is journal paper, that is molecular biochemistry or something. Yes. I forgot the name of the journal name. So any doubt? Yeah. What is that?
There are 2 types of experiments.
Ah.
Here you are showing only 2.
I am showing only 2D.
Method 3D also there. That is moderate.
Yeah, another 3D that you have to do because you are a biochemistry person, right? So you give me data, we will give you some models. We will design a model.
Moderate descriptors having 1600 plus descriptors are there. Moderate.
Sorry?
Use the moderate descriptors. So what is the this 2D we need 3D. 2D descriptors and 3D are the different. we are using here 2D only.
Yeah, we are using 2D only. What the paper is there, just we are reproducing the results, nothing.
One more note, and this 90 somehow having toxicity. Suppose some molecule having toxicity. How will you solve that problem?
Constant in the sense?
We will get toxicity molecule output product.
No. What is the meaning of constant here?
Toxicity.
No. Meaning of constant. You are saying that constant, right?
These 90 molecules are saying. These 90 molecules, one of them having toxicity. Suppose. So that toxicity molecule will produce another toxicity one. So how you rectify this toxicity before?
So that biochemistry part I didn't know. Slowly I also will work with biochemistry person. I will come to know. So if you want some doubt, if you have any doubts in machine learning part mostly, you can ask me, I can tell. Right? So you give me some data sets, I also will see what kind of data, and I will ask you some more doubts. Right? So since you are a biochemistry person, a research scholar in biochemistry, so I will ask more doubts, so while we working.
And we want some solution for that. So we are doing the software, right? So is it predictable if I engage with I want a result like automatically using AI? Like the pattern is different, like just like answers. Just as we are discussing, the DNA changes. So it will automatically calculate and give the solution of this molecular scenario, or we need to do coding.
Yeah, coding means the model we are applying, right? So model means, see, one model you take MLP.
In fact, in the molecule, right? The molecules are which are different right now. So
Maybe you can talk to sir during lunchtime.
we need give the next calculation of the next
No, actually, we are talking about machine learning part or biochemistry part. I am not understand.
So machine learning, can we predict the next the scenario that?
Yeah, so first let me tell you, in machine learning, what is that? Prediction part is different thing, training part is different thing. So that's what I told, right? There are some validation techniques, right? So the data you have, you split that data into some folds, right? Now you train the model. So you build the model with the help, just using all the training samples. You build the model. Now you take that model and predict it, any unknown sample, right? Now you have the data. So apply those techniques, nothing else.
Thank you very much, sir.
Okay, ma'am.
Actually, there is gap in our chemistry. We are trying to link whatever we know with the code that sir has written. So you give the input, and the input is related to a molecule, say your SMILES coordinates or whatever they are. You give the information to the program is run, and it will give you the descriptor values, right? It is validated. If the descriptor values are compared with experimental data, if the experimental data there is a deviation, then sir will go back and rechange his program code so that you'll get a better result, and it is more to the realistic value. So that is it. Thank you very much, sir, for the wonderful session, and I think there is a gap for us we need to learn more about this. Thank you.